I think this is actually perfect. Thanks, Alex, that I'm allowed to talk after you, because we do very similar things, but with the other method, or with one of the other methods, which is NMR. I tell you something about fragment screening. I work at Frankfurt, but this research is conducted in the context of iNEXT-discovery. And this is also an advertisement, so you can come to us and we can conduct experiments just as you can do X-ray screening. So iNEXT has started February last year, will run for 4 years, and it actually combines NMR, EM, and X-ray. This is where the acronym comes from. It's actually coordinated by Tassos, who chairs the session, and it's about structural biology. And if you want to come to us, you have to write an email to Christian Richter, but you find him or find me. Anyway, so we have a ton load of NMR spectrometers. So in NMR, you buy lots of these magnets, not just one beamline, but you run thousands of those, and they come, and that's probably more important here, with dedicated equipment. And in the context of fragment screening, you sometimes want to screen only fragments containing fluorine. This is NMR favorable, so we have dedicated fluorine probes. We have also very small probes needing very small sample. So this is something you typically can't have in every university. So this is why this is actually very nice to have these dedicated facilities. Now we also have invested in screening technology. This is being constructed by the company Bruker. You see robots that pipette samples. So we can run up screening campaigns up to 1,000 compounds in something like 3 days, with support by this robotics. This is all linked to software, both by Bruker and also written by us. And then you basically have these pre-temperature calibrated settings, such that you can run very rapidly in a pre-temperature calibrated system your experiments. This is actually quite interesting. In the fragment screening campaign, it may be beneficial. There are other experiments where you really want to have these samples preset. And then this goes on. You have automatic loading of your racks into the computers, and so forth and so forth. I want to talk to you about these fragment screening campaigns in the context of COVID-19. So I'm coordinating the worldwide network of all NMR spectroscopists that try to do something for or against COVID. And my stuff will focus a little more on RNA. This is the other molecule the virus has in addition to the proteins, but you see also common schemes. So in March last year, we started this network, which is now more than 200 people in 16 countries. And very simplistically, we said there are something like 30,000 nucleotides in the RNA genome and something like 27 proteins in the proteome. And so why don't we look at all of those and make them NMR doable? And once we have defined their NMR spectra, made the RNAs, made the proteins, then do screening. And we screen with the library that Alice has just introduced to you. This is this DSI POISE library. So each and every RNA and almost each and every protein is being screened by NMR with this iNEXT library. And then comes follow-up stuff, which I also explained to you, because this is also nice that the question came up. So you see, again, the architecture of the molecules of the virus. Very many of you will know the protein part. This is the 16 NSP proteins, then the structural proteins, 4 of them, and accessory factors. Some of the functions of those is not so clear. And that is mapped to an RNA, a genome architecture, where you have these stem loop structures up to SL5, then you have the start codon, you have an open reading frame 1A, which goes up to here, so it expresses up to NSP11, and then in order to actually also express the other guys, NSP12 to 16, you have to have frame shifting, and that frame shifting is under the control of a pseudoknot that comes up later, and then comes the 3' UTR with additional factors. And many of these factors are then involved in protein-RNA interactions that actually then mediate virus life and propagation. So you see here our NMR deconstruction of the virus, and we use a very similar approach as X-ray, and some of our constructs have actually greatly benefited from SARS-CoV-1 research, where we actually split all these large protein chains into small pieces and then look at them one by one. And so in a paper that we have submitted just before Christmas, I show you this is something like more than 30 constructs of the protein. And basically you see, but for the white spots, we have covered the proteome of the virus. And the same is true for the RNA. We have decomposed all the RNA elements, determined their secondary structures by NMR, and mapped this with a DMS footprinting to see the structured parts that can be drug targets in the RNA genome. That we were already able to publish. You see, it's like 50 author paper, 4 different institutes worldwide, Weizmann, Stockholm, and others. And I'm now going to tell you something about the screening, which is here now summarized. These are the hits, and I go through this in more detail, from the fragment library DSI that bind to the RNA elements. So this library has been developed, and it actually uses these fragments. And the idea of fragments to target proteins or RNA was originally developed by Steve Fezzik at Abbott using SAR by NMR, saying if I have 2 things, I have to see where's my camera. If this binds, sorry, if this binds and this binds, if I put them together, they will bind. And then in this great library, these fragments have been deconstructed such that we have synsons. And this is really nice library. And also in Frankfurt, we have this library at stock. So if you want to conduct your experiments, we have the library and we will conduct with you your experiments. You come with your target, we would conduct the experiments. So, I'm sorry. We have now also different vendor specs because we don't have the context anymore with enamine for reasons that are unknown to me. So we have this library again assembled, and we actually screen the library in fragments, 12 fragments per mix. And each of the library has 4.2 millimolar in DMSO D2O. And then we use 10 micromolar of RNA, 200 micromolar of ligand to screen. So basically you need a large NMR sample to conduct these screens. Now, what I show to you here is one thing that we do by NMR, and that is control each and every compound in each and every buffer that we screen with. And so this is the NMR spectrum of this A3 mix. And you see that we have the NMR spectra of the individual components, and then this is then in blue the mixture. And so you find each and every of these peaks in the mixture because you know where they are in the reference. There are fundamentally 2 different ways of doing screens by NMR. It's either RNA and protein detected, then you look for shifts in the HSQCs, or you do ligand detected things. You can either do STD NMR or water loxy or relaxation properties. I'll show you what we see in a minute. This is actually shown here. So basically this is one binder, binder 3, there's another binder, binder 8. And so you see here the binder 1 has a large chemical shift perturbation upon adding the RNA. This is 7 Hertz. This binder is a non-binder. It only has a shift of 3.4 Hertz. And this has even a smaller shift. We have water loxy. This actually measures the accessibility of a site to water in the presence or absence of the ligand. So if a ligand binds, then the accessibility to water is changed. And so you see here a huge water loxy factor, and here smaller and here none. And then you see T2. So if the small molecule binds to the RNA, then its T2 time is way faster, so it relaxes faster. And so you see here, this is reference, and this is upon addition of RNA, and you see how important the line becomes. And these 3 parameters we analyze automatically and get CSP factors, water loxy factors, and T2-CPMG reduction factors. And if 2 out of the 3 are above threshold, then we say it's a hit. And that's the hit of all the RNAs of the virus against the DSI library that we share with our friends in Oxford and with Tassos and others. So you see, also we see hit rates, similar relatively high hit rates for the fragments. And now comes a step that goes into science. What do you do with the fragments? What the heck do I do with a fragment? But to see that we can, in principle, target the RNA structure space of the virus. Now, we looked into the library, and the library by itself has a fundamental architecture. And this is at 1 functional unit. This would be here, this guy here, a benzene ring with a chlorine, and a second functional unit. This is this isoxazole, this 5-membered ring. And then it has a linker, and that's an amide linker. And so we see that we have classes of compounds that bind. And also interesting is the quality of this library, because we get specific binding motifs that bind to RNA. You take this guy, which has nitrogen here and a methoxy group here, and that binds. If you get rid of the methoxy group, you don't get binding. And if you add here or you shift this nitrogen, you don't get any binding. Or this guy actually binds to everything, including proteins. We also have screened proteins also overlapping with Oxford initiatives. So if you have 2 chlorines here, it binds everything. If you take 1 chlorine away, it doesn't bind anything anymore. So you go from binding promiscuous to non-binding with very small effects on the ligand. And so then taking this sort of already grown fragment, so 2 fragments are already fused in the library approach, we can actually dissect our chemotypes on the X and Y axis and see how often do they bind and to which RNA they bind. And now how to interpret this is really an art in itself. But I would like to now, in the last 2.5 or whatever minutes, tell you a specific project. And this is the guy, this is targeting the pseudoknot. The pseudoknot was already a bona fide target in SARS-CoV-1 because it actually shifts this NSP 1 to 12 to NSP 12 to 16 frame in this first open reading frame. So you don't make the polymerase if you don't get the frame shifting right. So it's really a valid target. And so we screened this, and these are the hits from the iNEXT library. I call it now iNEXT Generation 2 library. And then we actually looked over our RNAs. In this case, when we started, these were 5 RNAs ready. We had 168 hits. We looked in the vendors library, identified alike compounds, 4,400, did a selection process, boiled this down to 100 compounds, did a further visual inspection and boiled down to 50 compounds and did a secondary screen. And so the secondary screen identified these compounds, especially this one, and then compounds we knew from COVID-1, and we resynthesized this guy here. And there are others that actually are also, you know, could be nice, higher affinity targets. So you rescreen based on the initial fragment screen. And so from the 14 soluble ligands and the 3 synthesized compounds, we found 7 potential hits. And this DO1, this guy has the greatest binding properties. And then we redo NMR, and we see that even now at the ligand to pseudonode ratio 1:1, we get huge effects, first in the 1D spectrum, but then also in the 2D spectrum. So you see, not very well, but you see that there are black spots and red spots. And if we map the red spots where we detect binding onto Nenad Bahn's cryo-EM structure of ribosome plus pseudonode, then this actually maps out here binding site. So the compound, this guy, I mean, the chemists among you see that this is probably fluorescent, and it's in fact fluorescent. So we utilize the fluorescence and see that the DO1 actually binds with a KD of 2 micromolar. And we have taken this DO1 and do chemistry now in my lab to follow up on this DO1. And this compound is at 800 nanomolar. And you can imagine why I don't show you the identity of the compound, because at this point, after screening the fragment library, I generate new matter, and then I have to check whether I have to patent this, but that's ongoing. And we also have then established frame shifting assays, and we see that this guy, this second generation first fragments, you do 50 screens behind this, has a KD of 2 micromolar and inhibits frame shifting to 50% at a concentration of something like 20 micromolar. And with this compound here, we are now at biological testings. So with this, I hope I haven't run over time and would stop here. I have forgotten my acknowledgement slide, but I shall go very quickly back and show the people that are involved. This is the group in Frankfurt. And then there are way more, and we are spotting this one spot. So this is probably something of a 1% thing in our societies. Thank you for your attention.